start. Hi everyone. So today we have let's say an extraordinary. Oh, it started. It's morning in France, so it's a special day today. It's special timing because we have the opportunity to meet with Nicole Yuxi from New Zealand. Nicole is a doctorate assistant at the Institute of Marine Science of the Auckland University, and she will talk us about her works on microplastics, I guess burying behavior, ecosystem functioning, marine sediment, and maybe upscaling. I don't know. So the schedule is particular this time because Nicole is in the time. Session very late, very far from Europe and USA. So we have not a lot of assistant attendees today, but a lot of people are waiting for the recording to hear what Nicole can tell us. So welcome, Nicole, and welcome to the next park in the same time.
Hello, everyone. Thanks for inviting me to present my project and share my progress with you. I'm a PhD student from University of Auckland in New Zealand. My study focused on addressing the mechanism of microplastics influencing marine soft sediment, and I linked the observation to microplastics research and explored the broader scale shift on ecosystem functions. Because today I'm presenting from a remote time zone, so feel free to contact me if you have any questions, comments, or collaboration offers through my email. Well, microplastics is the most dangerous plastic pollution in marine environment, and its quantity increase with production of plastic pollution. Majority of plastic pollution will end up in the ocean and sink to the seafloor after exposure to rainfall, winds, UV lights, by weathering, and biofouling. If you type some keywords into Google Scholar, you will find heaps of research investigating the distribution of microplastics in the ocean, interact with marine fish and benthic invertebrates. However, we don't know the consequence of this interaction effects, as well as the impacts in particular ecosystem functions. While I focus on marine soft sediment, this area connects the ocean and lands. It accepts large quantity of nutrient inputs in either solute or particulate forms. It has key functions, including organic matter decomposition and nutrient cycling. While marine soft sediments are currently degrading due to multiple stresses, such as different types of contaminations, sea level rising, and climate change. Against all this background, I recommend we need a systematic research on how microplastics disturb ecosystem functions and quantify the loss of ecosystem services. So let's look into the real habitats, the natural seafloor habitats. Well, in these regions, networks among macrofauna, sediment conditions, and microbial community that shape the nutrient cycling. The environmental conditions, including mud contents, grain size, resources, the food quality and quantity determine the distribution of species in the habitats. In turn, the growth of macrofauna community shape the environmental conditions. Their feeding, burying, seasonal recruitment are essential for the temporal and spatial change of nutrient cycling in the habitats. The large macrofauna brings organic matter to the subsurface of the sediment that can be consumed by other meiofauna community and decomposed through bacterial process. While their feeding also control the growth of microalgae community at the surface that mediates the computational relationship with bacteria for dissolved nutrients. So the above feedback loop, as well as the networks, determine the nutrient fluxes, which we can measure from benthic environments. Well, previous research showed that there is a direct impact from microplastics on megafauna activity, mostly related to the solar movement, reduced feeding rates. Besides, some other research showed that microplastics can shift the composition of microbial community. Well, this study mainly focused on the individual toxic effects on individual components, whereas my study looked into how these combined effects really influence or interfere with network interactions as well as the feedback loop. And that will cause broader scale shifts on ecosystem functions. So the first questions I come up in my project is, can we see the significant change on ecosystem functions when microplastics change megafauna behaviors? Because the megafauna responses on the ecological level is not only the consequence of the stress, but also linked to serious change through the networks, as I mentioned in the previous slide. And this is not a simple questions, and I have a method for integrating mechanistic model to laboratory experiment to review the mechanism driver. Before we moving on, we can go through the experiment context. Well, I choose polypropylene as microplastic material, and I control the size within 0.5 millimeter. So polypropylene is one of the common commercial plastic materials that ends up in marine sediments. I mix this polypropylene particles with top 1 centimeter depths of sediment and incubate them for 2 weeks. So this pre-incubation step is very important for biofilling. And this step also can maximize the settlement of PP particles with surface sediment, allow microphytobenzols, bacteria interact with the PP particles, and that will influence the food resources for the megafauna. I have ambient and 3 microplastics treatments in this study. For the ambient, it's actually the fresh and clean sediment collected from natural habitat. I collect them from a relatively pristine area in New Zealand habitat. And then for the 3 concentration levels of microplastics, those were actually retrieved from previous studies from the mild contaminated sites in New Zealand harbors, Southern Ocean seafloor, and some heavily contaminated site in Hawaii Beach. I have 3 species in this experiment. I'll give more details in the next slide. This is Mandanid worm, Callinia bivalve, as well as the combine of these 2 species. Well, those treated surface sediment were then set up as the top sediment in the mesocosm. The species were introduced to the mesocosm as well, and I use luminol tracing methods to estimate their behaviors underneath the sediment through by 21 days incubation. Well, at the end of my incubation, I conduct chamber experiment to extract the overlying water samples between the sediment and the water interface. And this is for me to analyze the nutrient fluxes in dark and the light conditions. Also, I collect poor water nutrients, organic matter contents, chlorophyll A concentrations, as well as the observational particle mixing profiles, and I'll link them for further analysis. So firstly, we can look into how mechanical model work in my study. So from the view of transport action model, it actually creates a very ideal system, where bioexpansion is the parameter and the solar factor control the redistribution of organic matters through the sediment columns. This system simplify the sediment columns to two separate zones. One is oxic and another one is anoxic. Well, microbial activities, which is virtual in this system, was simplified as kinetic terms in the separated zones. So in this case, I wanted to know how perturbation as the main parameter that change with the concentration of microplastics. Well, the outcome from transport action model will help me to look into the potential consequence on the change of perturbations and link that to the nutrient cycling. Well, this slide shows the results from biodiffusive models. So after I feed particle mixing profile to the biodiffusion models, I can quantify the change of perturbation rate as a response to microplastics treatments. Before I show the results, we need to understand how species that I use in my project behave in the normal conditions. This is Mandanina 1, Macroclemanella. So this species feeds head down to the bottom of the sediment. The burrowing and the movement of this worm can cause a rapid subduction of fresh organic materials from the surface to the depths. This is the way for this worm to preserve their foods. And then they can suck up the fresh particles through the bottom and then release some waste or anoxic particles to the top. If you go to their habitat, you will find some volcano shape and anoxic sediment at the top layer. So this feeding activity actually creates a deep mixing profile of the particles through the sediment column. When I look into the perturbation rates, actually increasing the concentration levels of microplastics reduce this perturbation intensity. And this reduce can be referred to the loss of their deep mixing behavior. While another species is Telenida biva, Macromona liliana. It is also a deposit feeding biva. It usually inhabits within 6 to 10 centimeter depths of the sediment. It has two siphons. The longer one can reach out to the surface of the sediment or sediment water interface to grasp the fresh foods into their burrowed zone. And they have a shorter one to release the waste to the shallower depths of the sediment. So this feeding activity creates a random diffusive pattern on the particles. When I look into the results, there is no significant change on the perturbation rate. While when two species co-occur in the system, I found that the turnover of the particles in the sediment column depends on the species with a dominant functional roles. In my study, apparently the Mandanida worm was in a leading position to influence the results of a perturbation rate when in response to different concentration levels of microplastics. But there's still some possibility that upward and random diffusive effects on particle transportation might be a trade-off effect on vertical scale. Also, there might be the trade-off between negative impacts of microplastics and the interaction effects between these two species. So after we know the trend or the change of perturbation rate, we can upscale to the transport action model and to see how model estimates the results and the consequence on nutrient cycling. So with the loss of deep mixing or reduce of perturbation rate, there will be more particulate organic matter resources at top layers. More organic matter will be consumed within oxic layers, and that can equivalently increase oxygen consumption rate. So this graph actually shows the model estimations based on the results of perturbation rate. And then because of a reduced on perturbation and also the change on organic matter redistribution patterns, we will see less nitrogen regenerated through the pathway of nitrification and denitrification coupling as a result of regulated effects from deposit feeders. So here's some implications from my modeling estimation. But we should keep in mind that real habitats contains benthic microalgae, bacteria, and sedimentary environment conditions as the feedback loop to determine the nutrient fluxes. I then need to use observations from mesocosm to show whether perturbation is a solar factor to explain the mechanism of microplastics influencing the ecosystem function. So this comes to the second question about mechanistic driver. Then first thing is we need to know how does laboratory experiment tell us that is different from the modeling system. We should regard the modeling estimation result as a result from the cost-effect relationship between microfauna behaviors and nutrient cycling, while the real habitats or the laboratory experiment actually contains multiple interaction among the components, as well as the direct and indirect impacts from microplastics on these components. Therefore, this cannot be simplified as a cost-effect relationship, but more related to the network effects as well as the feedback loop. Therefore, the comparison between the outcomes from modeling systems and the mesocosm is actually the comparison between a simple and a complex system. So this is the way for me to look into the mechanism driver. So this graph actually contains really dense information. But we can go through this graph one by one. Here I use multivariable analysis to evaluate how variables associate with each other to determine the change or the responses from the ecosystem to microplastics. So this is the cap graph. These are all the variables that are measured from the mesocosm. So this is the luminifold abundance within sediment columns, and I will explain this in the next slide. And this is the ratio of the particles with penetrated to the sublayers of the sediment, and this is the particles on the top layers. While I also include the nitrogen fluxes in dark and the light, ammonia fluxes in dark and the light, as well as nutrients within sediment and organic matter contents, as well as chloride A concentration. So I use all sorts of variables that I measured from lab experiment and to look into what are some dominant variables that can explain the change of ecosystem functions when we increase the concentration levels of microplastics. We should be aware of the location of these treatments on the graph. So, for example, this is the worm groups from the control treatments. And once we increase concentration of microplastics, it starts to move to the central part. The same patterns was shown on combined groups, apart from the fluctuations still moving down to this side. And this is about linear bivalve groups. Although they kind of accumulate in the same area, but there's kind of a downward chain for them to moving along this way. So based on this movement patterns, we can select some key variables. Also use Permanova to test if there's a significant impacts from microplastics or interaction effects between microplastics and the species. So the first variables that I selected was organic matter contents on sediment surface. So this variable has significant impacts from microplastic treatments. And actually, the general increasing trend was is very similar to the model estimation, as I mentioned in the previous slide. Well, the more important variable is the luminol abundance within sediment columns. And this was actually a result from the interaction relationship between microplastics and species. It means that the change of this variable really depends on the concentration levels of microplastics as well as the species within the system. And this variable followed with the change of particle movement along sediment depths, especially in the surface layer and sublayers. And the last but important variables are nitrogen fluxes in darkened light. Remember, in the model estimations, reduced nitrogen regenerated through coupling of nitrification and nitrification process was one of the indicators of the change of ecosystem functions when we lose the perturbation rate or with a reduced perturbation rate. We actually found this in the dark conditions when there's no photosynthesis that happens in the system. However, when we add the light into the system, which means that photosynthesis microalgae start to work on nutrient cycling, it start to release more nitrogen through the sediment system. And from this results, it actually shows that in addition to the change of perturbation rate, we need to think about other process, especially the process that related to interaction between microphytobenzols and the bacteria that will participate in nutrient cycling in this system. So let's talk about the driven factor and also the important factor that I depicted from CAP graph. The luminol abundance within sediment columns actually means the organic matter, particulate organic matter resources within sediments and sequenced by deposit feeders. So this graph, this image were actually the surface sediment when I stopped incubation. So this dark size without luminals on the top, actually the signal of the results of feeding activities of deposit feeders underneath the sediment. But we incubate the species with all sorts of components as well as microplastics through 21 days incubation. And then this can be the results from the network effects. And then there was a general decreasing trend. It means that less organic matter resource can be allocated through the sub-layers. And at the first slide of my presentation, I mentioned that one of the role for deposit feeder or macrofauna underneath the sediment is to transport those organic matter to the sediment and that can be used by either megafauna community or bacterial community. It means that this kind of linkage or positive effects from megafauna on benthic environment might not valid at this moment. But based on my laboratory experiment, one conclusion is that there is a broken regulation effect from megafauna on microbial activities, and that will cause a faster removal rate of nitrogen through the top sediment layers. Then we can compare our experiment results to the mechanical model to estimate the chain reactions. As we remember that modeling results emphasize the reduced perturbation rate with shift of organic matter redistribution patterns. And this makes more organic matter consumed in aerobic process. Well, when we integrate the lab experiments, we found that actually, potentially, the interaction effects between microplastics as well and the microbial activities also integrate to the response of ecosystems to microplastics. So these two processes actually happens at the same time, and that will influence the ecosystem functions in the habitats. I also have extended experiments based on the result from the first two chapters. Because the first two experiments, there's no significant change on perturbation rates in telling bivalve groups. However, my early study in 2019, as well as a related study from Julie Hope, and she was one of my supervisors at that moment, we all found there's a reduced capacity for telling bivalve to reburrow themselves into the sediment after 30 days incubation with microplastics. It probably shows that telling the bivalve has different mechanism that is different from antennate worm, which should be considered for the risk assessment. Therefore, I have 42 days incubation to allow telling bivalve interact with the same concentration levels of microplastics through 42 days. And I use pressure sensing techniques to monitor the pressurization behaviors during the incubation. This allowed me to check if they are still alive during the incubation. And then after this incubation, I have 3 hours thin per experiment. And also this allowed me to detect critical pumping activities after they recover from the incubation. And then their behaviors, their pumping behaviors actually cause the periodical exchange of oxic and anoxic pore water through sediment and water column. So these activities can temporarily oxygenate the sediment environment, especially the barred zone. And this behavior was actually stimulated by Niels Verkenberg in his previous study. While there's some studies that recorded in Michael Thompson, so that will help me to understand whether there's some critical change on their behaviors. Then when I fit those pumping duration to transport action model that refined by Niels Verkenberg, I found that in the low concentration treatments, the pumping duration of telling bivalve can no longer oxygenate the sediments, regardless of sediment types. So these results have way more important implications, and that should be integrated to the results from the second experiment. Well, based on my 3 chapters, here's some summarized key points. First of all, the shallow penetration patterns of particle resources that can be the mechanism driver, especially for the habitats that dominated by deep mixing or deep burrowing species like the Mandanio worm. Well, in that case, more organic matter that consumed at top layers will change the pathway of nutrient cycling. And we probably will see the reduced nitrogen that produced through the regulated effects of macrofauna. But because of the interaction between photosynthesis, microalgae, and bacteria, and also the stress from the microplastics that cannot be excluded from the real habitats, there will be a faster removal rate on nitrogen gas through the top layers. So here's some implication that I summarized through my project. We usually found lots of studies when it comes to the microplastic contamination or other types like heavy metal, all sorts of stress. We mainly talk about the toxic effects on single species or single components, whereas my study actually showed that the impacts on the feedback loop through either traffic interactions or some regulated effects from macrofauna really matters. And this can cause severe impacts when compared to the single toxic effects. While another point is that it's really hard for us to measure the concentration levels of microplastics in the real soft sediments. And that is mainly limited to techniques or it's a kind of a time-consuming work. However, from this experiment, it shows that the low concentrations of microplastics, probably you can find it in the real habitats, really cause the impacts on macrofauna behaviors, including the reduced deep mixing behavior, as well as the reduced oxygen availability due to the lack of pumping behaviors of tennant bivalves. Therefore, the bigger impacts might be related to the low dosage. The third point is that we know biodiversity is the fundamental base for maintaining nutrient cycling and ecosystem functions in benthic environment. And we need to integrate the macrofauna behaviors to the risk assessment. And this is an urgent need. So thanks for listening. And this is all for today. I'm happy to receive any questions, advices, or collaborations from you guys, and feel free to reach out. Thank you. Any questions?
Okay, thank you, Nicole. Thank you for the one who attended today. Any question to Nicole? If there's not, I will go for common question.
Yeah, feel free to
Okay.
ask me.
Okay, thanks again, Nicole. Okay, very interesting, and I like your different conclusion when, especially when this important point you raised that even in low concentration, we as you had some effect on the activity and on the fauna and then as a driver of the cycles you impacted the nitrogen cycle. I have two kind of specific question about how you could explain that even if you have by example by the pumping activity of the bivalve, even you had an impact at low concentration, and then the impact was lower, even if you have an impact compared to the control, was the impact was lower when you increase the medium and high activity. Do you have an idea how to explain that? Yes, on that graph.
You mean why they have a really low pumping duration when there's a low concentration levels in micro?
Yes. And then
Um,
that the duration was higher then.
yeah, I think this is a really good questions about why it is a low dosage. I did some research about the additives of plastics. So I think that the additives of the plastic materials that contain some endocrine disruptor. So some previous lab experiments show that this endocrine disruptor will cause some low dosage impacts. However, the PP material was claimed to be additive free or less additive or less toxic for human. However, I still see the low dosage impacts, and this is not the first time I saw the low dosage. When I was doing my first experiment, that in 2019, there is a low dosage impact on reboral capacity of this species as well after exposed to the microplastics. So this is the one explanation. It's about the additives in the plastic material. The other reasonable explanation, probably because of the interaction between microplastics and the microphytobenthas as well as bacteria was really strong when it comes to the low dosage. Maybe it influenced the bacterial activity as well. And this means that in low dosage concentration levels, either the direct impacts from microplastics or the indirect impacts from the change of environmental conditions will pose a stress on telling the fiber. That's maybe another reason for them to change their behavior significantly.
Okay, thank you. But that's why that microplastic is a tricky word. When you have that additive stuff, also that we say, okay, we have the plastic, but also we have some different additives and also different microplastics.
Yeah,
It's a,
shape,
it's,
size, concentration, all
yeah,
sorts of, it's kind of combined and really complex context issue.
yeah. And by example, what was the shape of your microplastic in your experiment?
So let me show you with, sorry, it's in here. It's actually, this image was actually recorded under the microscope. So there's no specific shape for them.
Okay.
Because I ran them through the coffee machine. It's a coffee grinder. So this is kind of a diverse shapes within one scoop of microplastics. But I control the size of microplastics to make sure that they are kind of in the same range of the size.
Okay.
You regulate the shape, I would say. This is
Yeah,
just
but
a
it's
small.
not okay. It's not dominated by fibers or by protein.
No,
Okay, it's
no.
no, okay,
There's no fiber in this. It's all about fragments.
okay, okay. Interesting. And if I can ask another question, if nobody wants to do that, no, not yet. Then it's okay. We've been talking about the ventilation part of the bioturbation, the previous question. If you go to the particle mixing part, also can see that you state in your conclusion that it's a complicated world and we have to take account the behaviors of the animals. You're right, because if you look at the luminophores at the CDMO working in regard to ventilation, we can see that the effects are different, and that also what we have to take into account, especially when we look at the cycles here, the nitrogen cycles, that the two components of the bioturbation, they do not react the same. Sometimes they do, sometimes they don't. And it's very important to look at both components when we look at cycle as you did, because here the result with the luminophores were different pattern that what you had, I say, for the bivalve, for the ventilation, the pumping. But the worm also, you have that medium, you had that medium aspect, medium effect that was higher than the lowest, the low concentration, high concentration. But for the bivalve, oh no, bivalve look almost the same pattern that you have with the pumping rate. No, it increase and then slow.
It's actually not really significant.
Yeah,
Yeah, it's the significant only between the high and the control.
and the control.
Yeah, but not really significant because I think when I collect the sediment samples through different layers, I found that it's probably mainly because of the burrowing and the feeding modes of the bivalve. Because when you look into the animations that I show in here, in most of circumstances, the particles was just accumulate on the top of 6 centimeter depths. While for the bivalves, I can found the particles that even down to 12 centimeter depths or 10 centimeter depths. This is just how two species work with the particles. However, I think it's more related to the mechanism or the main functional role of this species in the sediment. Based on the literatures, most of the literatures about the mandanite worm, while there's kind of limited literatures, is more related to their deep burrowing and the ability for them to transport the surface particle to deep layers. While for the telling bivalves, not only the particle mixing, but more related to the pore water regulation, which is the bow irrigation. That's why I start to think about, okay, if there's no really significant change based on the particle mixing profile or luminophore abundance that I collected from the sediment columns. However, there's still some change that are based on previous study on their reborrow capacity, then why this happens. Therefore, I start to think about, oh, it's more related to their bow irrigations, because once they reach out their siphon to the sediment water interface, they can actually spin on the water column to some point. And then this kind of really active activity is really kind of a reach out, searching for food, spin for a while, and reach back and pull it back. So this activity actually keeps oxygen penetrate to the sediment in a periodical way. So I don't think that if we can't see this really significant impact on cultivation rate on tailing bivalve, it means that the tailing bivalve was not impacted by microplastics. It's maybe because we didn't measure it. We should measure the right indicator. That's actually one of the implications that I integrated in my final chapter in my thesis. It's all about what we measured and what are some indicators that really related to the response of megafauna to the stress. Yeah,
Yeah, that's
yeah,
you're totally right. The proxies, depending on the proxies we use to measure, at least at that time by automation, it could change the
yeah,
result,
yeah,
but you have to get the whole picture to understand.
yeah.
Adrienne has a question.
Hi, great talk. I was wondering why you chose the intervals that you did for like the sediment intervals for measuring the luminophores, because what it looks like is that you have so few intervals that it's really hard to resolve. If you had, more varied transport, it's hard to resolve uh, whether or
You
not
mean,
how well it matches the prediction.
yeah, you mean the interval between sediment samples or interval from
The
the
interval
model?
between sediment samples. So clearly, like in the first 4
Um,
centimeters, you only have, for each of the different treatments, you only
yeah,
have 3, right?
yeah, this is actually practical questions that I'm just waiting for someone to ask me. It's really, really hard to analyze the particles or recover the luminophores from sediment samples because I have several replications and different treatments. Then let's back to that profiles. And then I choose 2 centimeter. It's mainly because I can use 2 centimeter as a kind of estimated depths for oxygen that can penetrate. So if I can collect or recover or estimate a luminophore penetration through the oxide layer and sublayers, that I can easily input that to the model. So I only want to look into if there's more accumulation on oxide layer or anoside layer. But actually, I would recommend to have fine layers, especially like 1 centimeter by 1 centimeter. However, it's not the practical and efficient way to do the laboratory experiment, especially if you need to recover those luminophores under the microscope and examine the numbers, content numbers of luminophores under the microscope, and they convert it to the image and sent to the MATLAB. So this is kind of a very long-term process to analyze each samples. But I know there's new techniques combined the image recording and the thermal experiments. In that way, they can record the movement of particles through the thermal, and then you can slice the image to very fine lengths or fine increment. So I think that's the better way to estimate the bioactivation rate for the further research. However, I think in my experiments, it's actually enough to explain the change. Therefore, I choose this increment. Yeah.
So if you were basing it on the oxygen penetration depth, did you actually look at each of the cores at the end and look at where the
Redox.
redox discontinuity was in order for you to determine that 2 centimeters was where
Yeah.
you wanted to do it? Or
Yeah. Not only have a look on, because we know that line is not just evenly underneath the sediment. It's kind of in this shape. It's kind of randomly in your incubation or in your buckets. And also it also have a spatial heterogeneity. Therefore, I need to retrieve some values from literatures and also think about the very, very ideal systems. Just assume, okay, 2 centimeter depth is the maximum depth for the oxygen can penetrate for some cases. And then to look into if there's some accumulation or there's some different patterns through through the sediment columns. So it's really about the estimation, and the only important thing is to look into the differences from the ambient and the microplastic treatments. That's the focus of my experiment when I fit those particle mixing profile to the model.
got it. Thank you.
Of course.
Any question from the attendees? I do. Maria, Marta, no? No question from you guys? Okay, so Nicole, thank you again for giving that talk. It was very interesting, and thank you. Okay, we have some difficulty to set up because of your place from New Zealand. So not a lot of attendees today, but people, as I told you before, they asked me to be sure that we record it. Then it could be viewed on the NearSpark channel. And I hope you will use the network also to, after your PhD student, to maybe try a postdoc position. Do not hesitate to either
Sure.
you, either the others to contact Nicole to
Yep.
discuss with her about that and also about our fresh results. And I will say good night for some of us. At the end, thank you for being here today, and have a nice day, the others. Okay, thank you again, Nicole.
Thank you. Good day.